And thanks, Ben, very much for the invitation and for setting the schedule. It's been delightful. This might be my 6th straight hour of talking, and so I'm going to be, but I'm still going strong. So I'll re-advertise what Tom just advertised. This is all in all its glory, a book with Dalton Conley, who is a PhD in sociology and in biology. So I'll cut right to, maybe I'll come back to the book a little later. So I want to cut to the main part of the talk today, which is to go back to a very basic model of a statistical model of intergenerational transmission, and partly just reflect on how people and how scholars interpret something like beta 1, which is just how similar are children to their parents. And similar here could be based on their income over their whole life, or their education, or some other measure of socioeconomic status. So this type of regression is done across many disciplines, and there's comparisons made across countries, across states within a country, across time, and so on. But they're of this typical flavor where you're trying to link children's, children with their parents' outcomes. And then the question is, well, when you have a strong link, what are the ways that you might interpret it? And so I've listed a few here, but broadly, you would say it's some combination of genes and environment. What are the things that parents pass along to their children? It's both their genetic inheritance and the environment that they're raised in. So it's not surprising that beta is some combination of that. And then the work I'll be talking about today is to investigate this and think about how genes and environment might interact to produce the outcomes of the next generation. And part of what I'm going to be talking about, you've seen in past presentations, which is just that there's from the biological genetics and bioinformatics literatures, there's new ways of measuring part of this transmission, which is what is passed genetically between parents and children. So I'll go into that a bit today. So something of the blueprint, I'm going to have a few slides on genetics reminders, because I'm going to be talking about data that is that has a large amount of genetic information in it, so it's more of a reminder. I'll discuss a bit, going back to the book, what we describe as a major paradigm shift and some of the implications of this paradigm shift that's happened outside of social science, in the natural sciences or the biological sciences, and thinking about how genes work. In particular, how big the effects are for individual genetic variants. And the short answer of how then social scientists are going to use what this paradigm shift, the implications of this paradigm shift for social scientists, is to accumulate information across individuals' whole genome and sum it all up to what's called a polygenic score. So I'll describe that in a slide, and then go into an actual analysis of real data using a nationally representative study in the U.S. I'm open to questions and answers throughout it. If that's not what's typical, I'm happy to be interrupted. Okay. So the couple slides on genetics is just to get the scope of what. If you have a data set that has human genetic material in it, what does that look like when you open it up on your computer? Is in some sense what I'm going to walk through. So human DNA is approximately 3 billion letters long, or nucleotides long. So your data set could be, for each person, 3 billion letters. Okay, so and what I'm going to describe is a subset of that, but you should think of that as a starting point for the size of a data set that would have a complete survey of the entire genome. These letters, you can think of them as being accumulated into genes on top of chromosomes and so on, and then some folk definitions of what these things are. Genes provide instructions for building proteins that in turn do something inside your body, have some kind of effect on your body in the form of proteins or so on. But and should also note that the vast majority of the locations, there's no variation in nucleotides. And many of you know this, that if you take two people, 99.9% of all these letters are the same across any people in the world. So that's a really important first note. So it's only the leftover 1%, 0.1% or so that we're even going to be focusing on. So if you had a data set that had 3 billion letters for every person, almost all of those 3 billion letters are identical for every person in your data set. Okay, so that's one reminder of how similar humans are. On the other hand, we're going to be focusing on that small amount of all the letters that do vary across the population. One unit of analysis in these genetic files is a single nucleotide polymorphism, which is just a single letter difference or change across people. The shorthand is SNPs. So of those 3 billion letters, a small proportion actually differ across people, and that's what we're going to focus on. The convention, when you come to a point in the genome where people actually have different letters, is to say, well, the letter that's more frequent, we're going to call that the major allele, and the one that's less common is the minor allele. That's just the convention. And then one way to look at your data is to ask, at each location, for every person in my data set, the genotype for that single letter change or for that single letter variation in my data set. One way I could actually quantify that is for each person, I'm going to have either 0 of these rarer alleles from each of my parents, meaning both of them gave me the common letter. Maybe mom gave me the common letter or dad gave me the common letter, but not both. Or both of them gave me the common letter or the uncommon letter. So this is both showing that you have 2 parents that both contributed one of those letters. And then for each person in your data, at each column of data, you can ask whether I have 0 of these rarer alleles, 1 or 2. So when you actually open up the data file, you can imagine having hundreds of thousands or millions of columns of data, which are just 0s, 1s, and 2s. So that's a lot of data per person. That's hundreds of thousands of numbers, 0, 1, and 2 per person. And then how that's actually summed across all the letters to get a polygenic score is to first envision an approximation of a true model, which is you have some outcome like educational attainment or height, and the genetic contribution across the whole genome to height, you could imagine summing up each of the individual contributions. So you have hundreds of thousands or million of these individual X's, each of these individual contributions that are across the whole genome, and each of those contributes a little tiny bit to your height. And one way of capturing the entire genetic contribution to your individual height is to sum up each of your 0s, 1s, and 2s multiplied by how much of an effect on height each of those SNPs have, each of those variants have. And then when you add all that up, we can call that a polygenic score. So that's where I'm going. So the paradigm shift I want to highlight is, in the last 10 years or so, and we go through a lot more detail in the book because it's had a lot of implications for social science, is that I'm going to call the old paradigm the simple version of the old paradigm is one gene, one disease, and potentially one drug, which is there's a lot of health outcomes that might have genetic that have strong genetic effects on health outcomes or disorders. What we want to do, this is the old view, is that we just need to go across the genome and figure out where in the genome there might be an error of some sort, say, that's going to have a high impact on an outcome. So many of you have probably heard of BRCA1 or BRCA2 variants that put women at high risk for breast cancer. That's an example of one gene, one disease, and potentially eventually one drug that would reduce people's risks. So there were some early successes. APOE is a success in a single or a small set of genetic variants that have a high increase in the likelihood of Alzheimer's and dementia. So those are successes of the old paradigm where a small amount of genetic information might increase the risk for certain outcomes like breast cancer or Alzheimer's by a lot. So then the question with these initial successes is, well, maybe that's true for most things that we care about. Maybe it's a matter of finding in the genome the 1 or 2 places that really matter for smoking, or that really matter in that case for test scores. So that's, you can see how that's stretching it. But that was a possibility. There was these big effects for certain types of cancers and other health outcomes. Maybe there's something similar for social outcomes. So that turned out to not be true. After these initial successes, really almost nothing else. There's very few other examples where a small amount of genetic change across individuals increase or decrease your likelihood of having a cancer or some other health outcome. So that's what I mean by an old paradigm. There were some successes, so it's not completely untrue. But what's turned out to be more true, or more likely to be true, is that instead of one gene, one disease, one drug, there's many genes, each with a potentially tiny, tiny effect. And that really transforms the whole type of analysis one would do in trying to characterize a genetic score or a genetic risk for certain outcome, for the outcomes of interest. So as an example, the top hits for smoking, the top single letters that matter for smoking contribute some amount. Some people would call that a very minor effect, or very minor amount of variation. The top hit for obesity has an amount of variation in the population explained of 0.3%, not 3%, 0.3%. So those some people would view as modest or maybe small. And then you go to something like a social science outcome like educational attainment, and you see an order of magnitude reduction in the biggest effect. Okay, so this is what I mean by a paradigm shift of not only are there not one gene, one disease, one drug, that the top hits are tiny, 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 tiny. And that's in some sense why, that's partly why I'm going to describe the new phase of measuring genetic effects on an outcome like education, which is to sum up everything across the whole genome, because the top hit basically doesn't matter. Okay, so this had some consequences. So there was 5 or more years in the social sciences where this old paradigm was pursued, and you get results like, so I've taken from many different disciplines, you get headlines of 2 genes predict voter turnout, or a single gene affects delinquency in a small. So what's happened here is that, so these are all not right. These are all, what's happened is that these are all false positives. We know now, in hindsight, that the top hits are tiny. So in order to find something really tiny, you need a really big sample. These are all really small samples, and they find really big effects. So these are all false positives. And I just showed that, economics, criminology, sociology, and political science so this is a top journal in political science. This is one of the top journals in sociology. So this had some ramifications across social sciences of pursuing this particular paradigm, which is that I need to find that gene that really matters and then predict something important from the social sciences like voter turnout. So that's turned out to be probably a dead end, and probably has had dozens of papers that have turned out to be wrong. So I'll bracket that and say now there's a different approach which doesn't have the same downsides, probably has different downsides, which is to use what are called genome-wide association studies. So instead of the old days where you thought there was a small number of genes that really matter, now it's just throw up your hands and say there's no big gene effect. There are only small gene effects. So let's go find them computationally. Just saying there's something there somewhere. If I have 10,000 variants, I'm going to run 10,000 regressions. If I have 3 million variants in my data set, I'm going to run 3 million regressions. I'm going to correct my p-values for having run a lot of regressions. But otherwise, it's just a data mining exercise to see if I can find something there that replicates across studies. So you can see how different a paradigm this is between something like BRCA1 or APOE, where you think there's this causal mechanistic understanding of gene to protein to something that affects your brain or something like that. Now that's all thrown away, and it's just it's a data mining exercise. So back to this. Here's the data mining exercise again, which is you have an outcome like educational attainment. You're going to run hundreds of thousands or millions of regressions and ask if I can find places in the genome where there are small differences in educational attainment based on having an A or a T at a certain location. So this is not a perfect representation of the truth, I'll outline two issues that you probably have in mind with running regressions like this. You might use different language. One you might use is confounding, which is that when you run a regression across a genome and you pool people from all over the world together, you might come up with something that you might call confounding and that geneticists call population stratification, which is that across the world there are places where you're more likely to have an A at a certain location and other places in the world where you're more likely to have a T based on the history of our species only. And that frequency, that difference in allelic frequencies, you might actually find that showing up here. So if in Louisiana you're more likely to have an A than a T, and the school system in Louisiana is not as good as the school system in California, you might find a lot of Louisiana genes here. But that's not what you want. So what the current fix is to control flexibly for the rest of your genome. Basically, you take all the other SNPs and you add them into a principal. You take a principal component analysis of them, take a bunch of factors, something like 10, and control for quote unquote the rest of your genome as you're running these regressions. So that's the current fix of this problem. The other problem is that you're expecting tiny effect sizes. An implication is that anything you might have found a few years ago are probably wrong because the sample sizes were too small to find something that's small. So the fix here is to err on the side of huge samples. So if you have measures that aren't so great as an outcome, that's okay as long as you have huge samples. So what I'm going to show you are studies that have hundreds and hundreds of thousands of individuals in order to find these really tiny effects. So these betas, your expectation are these betas are very, very tiny. Yeah?
So if you have an effect, so if I have sort of secondary effects where I have a gene that makes me stupider if I'm in a hot humid environment, for example, and then so that I have sort of a genetic thing that then is affected by an environmental thing that couples to a social outcome.
Yeah.
How is that getting handled?
So you're saying that there might be important gene-environment interactions between, right? So I think the answer to your question is what you're going to pick up here is you're going to, so go back to this point, in order to get really huge samples, you're going to be pooling data from many parts of the world, mostly in Europe and North America, but not only in Louisiana, I guess is the point I want to make. It's from all over the country or in other parts of the world. And what you expect to find are instances, betas, that survive, that are true across environments and not particular to some environments. If one beta only works in Louisiana temperatures, then you're probably not going to find it unless you have hundreds of thousands of people from Louisiana in your data. So there's several. I'll come back to some more implications of that. But the first punchline is that the betas that you think that you actually can detect are likely those that are as I'm going to say true or are operant in many environments, not just a small subset of environments. Yeah?
Are you also considering interactions between genes? Like I know that gets a huge amount of possible combinations.
Right.
But also
In these versions, no. But that's something, so the reason that can't happen now, when I say now, I should say like a year before now, because we're right in the middle of a major change in what I'm describing. So I'll say retrospectively, the reason you can't do that is that the data sets that make up hundreds of thousands of people in total have been typically from 70 different data sets all mushed together. That's not going to be true forever, but in what I'm going to show you that was true. So it's even worse that what you'd like to do is run a regression with all the X's and at the same time, and maybe all their interactions or something. But you can see that with hundreds of thousands or millions of X's, you don't have that many people. So you have some sort of data reduction strategy. So this is not a particularly sophisticated data reduction strategy, but the one that happens are these principal components. You take all the rest of it, you jam them into 10 factors, and those are controls over here, and then you look at what the effect of that one variant is. So, and to repeat what you said, which is true, there's a linear approximation assuming additive genetic effects. That's not going to always have to be true. I mean, the evidence that I've seen suggests that this is pretty good, but we just don't know the answer to whether there's some lurking, really important gene-by-gene interactions that are just not going to show up in what is done now. Other questions? Yeah.
Can you speak more to how we might think about what genetic effects are?
So it's a great question. I think maybe I'll just answer it by saying I'm not going to be able to answer it fully, because I think you're asking whether we're picking up some kind of causal effect that has some clear mechanistic set of effects where if you have an A versus a T somewhere, that means some kind of protein has changed, and eventually you get to something happening in your brain or body. That's, I think, what you would prefer. What you get out of this is just, on average in your data set, are people with A's versus T's going to school a little bit longer? And it could be because there's a mechanistic underpinning of that, or it could be because what you're actually capturing is a letter that's related to height, and that tall people seem to do better in school than short people. That you can't decide which one of those two things are true. Other questions? So when you accumulate these hundreds of thousands of people, you're going to then run all your regressions, and so now you have hundreds of thousands of regressions, meaning you have all these betas and standard errors, and then the convention is to present them in a way that looks like this, which is called a Manhattan plot, which you take all the output of your regressions and you line them up to where on the genome they were, so you're ordering them by chromosome, and then tall buildings here are good news, which is these are signals that you can actually get through all the noise after you've corrected all your p-values. So here's the line that says anything above this line is statistically different than 0 after I've made the correction that I'm running a lot of regressions. And you pick apart all these letters that are above this line, and you're going to call those genome-wide significant hits for education. But what's presented here are p-values, so small p-values are high buildings. Low p-values are all this background noise, and you're not sure if that's noise or the truth. And this is a sample of 300,000 people, and then by convention, you're going to take these top hits and ask whether they replicate in another large sample to see if to maybe further convince yourself that these are actual effects rather than luck or bad luck. So I'm going to turn. So this is one way of showing all the regression results. I'm going to now, in some sense, turn it on its head and say instead of ordering it by how small the p-values are, I'm going to order it by the effect sizes of the so now these are the betas. These are how important it is to have an A versus a T in terms of years of schooling. So the top line is a benchmark for something for height. So it's a different outcome that has maybe a stronger genetic set of genetic effects. So this is saying the top single letter change in someone's genome has an effect of about 0.2 standard deviations in height. So that's maybe important. You can see a drop off that the next 6 have something like a 0.13 or 0.14, and then another drop off, and then a tail. Then I'll show you the black dots are years of schooling. So the top hit has a really small effect, and all of the rest are even smaller than that. Okay, so in the old days, we might have taken the top hit and then used that in our further analysis to figure out why that letter seems to affect years of schooling. These are also small that what we're now doing with this information is to sum this all up into a single number called a polygenic score, and then take that to another data set as a single number instead of a whole bunch of effect sizes across the genome. Yeah. So I'll tell you what the convention is. There's no ground truth of what one should do. So now at this point the exercise is, there's a lot, so I think it's the next slide, which is you have some now researcher degrees of freedom. I have all these betas, and these are just these are the ones that are statistically different than 0 in the GWAS in this thing. These are just the top 74, but I have betas across the whole genome. I have hundreds of thousands of betas. So now as the researcher, you can decide, how do I want to sum that information to a single number? And some of the choices you'll make is, what fraction of the results do I want to use? I can use all of it, all the betas, or I can only take the ones that are above this line, or I can set a line that's slightly different and take everything above another line. So that's one choice, is how many of these betas you want to sum up. And then another choice is, do I want to treat this information the same or differently? I could weight everything the same, so that every one of those letters contributes the same amount to the summary score. The convention is to use the betas as weights, so the bigger effect sizes contribute more to the score than the smaller effect sizes. So the convention is, well, the convention that's established is based on the answer to the question, what's the way I can make these choices that makes out of sample prediction the strongest, right? So now it's a question of, I can do a lot of things using all that information. What choices can I make that out of sample will predict educational attainment with the most accuracy? So the convention now is to use everything and weight them, and weight the analysis, weight each of the SNPs based on the betas. But maybe that will change. That's less clear. So what I'm going to show you then is the next step, which is you're going to apply these weights to a new sample. I'm going to show you some information with what's called the health and retirement study. So this study is of just 10,000 people or so, where I have all their information, all their social science type information, plus their genetic information. I'm going to take these beta weights and apply them to the out of sample to the HRS and create a genetic score, or polygenic score inside the HRS that uses all the information in this larger sample. So the interpretation is maybe in quotes. This is the genetic propensity for the trait. In our case, it's going to be educational attainment. The advantage of doing it this way is that you have high out-of-sample predictive power. In our case, I'm going to have a single number that predicts 6 to 7% of educational attainment in the sample of HRS. So it's a single number that's this weighted average of all these letters across your genome. Other questions besides? Yeah. So the betas are captured by individually running a regression with the PCs controlled. So that's where you get the beta. So that's where you get your truth. So okay. Now, what I'm going to show you, maybe to jump ahead, is that I can now in the HRS I can also control for PCs, principal components of that sample. And again, that's partly by convention what one might do, because if you believe this is the truth, you might not need to control for PCs otherwise. But to answer your question, in the GWAS, all the PCs are controlled at each regression. And then in the out-of-sample part in the HRS, you can also control for principal components within your out-of-sample, within the HRS sample. OK, so some uses, you've seen Dan Belsky present one of the uses of polygenic scores somewhat recently, which is you take this score for education, which I'm going to use also, and ask across children's into adults' life courses, where in their life course or in what outcomes in their life course do we seem to have correlations between this score and early reading, and it could be smoking, it could be how long you go to school, it could be how much money you earn in old age. So that's one use. That's not going to be my use. I'm going to do something a little different, which is to use this as a way of capturing G or genetics in a gene-environment interaction study, where actually more interested in how the environment works in some sense. So I'm going to now show you what the environment part is shortly. So I'll refresh your memory on where we're on like the underlying model of inside this beta 1, there's genetics and environment. I've now shown you how I'm going to measure genetics. So there are not many economists here, so you won't be as maybe interested in these particular people. But in the economics discipline, one of the overall handbook chapters in how do you measure intergenerational mobility and what does it mean? From this, I've taken and made it and bolded and underlined. So in asking how do you interpret beta 1, the authors say there may be genetic differences in ability that are transmitted from parent to child, and that lead to intergenerational persistence and income. So this is my underlining and my bolding. But what I take from this is something in the background of there's a possibility that part of beta 1 has a floor to it. There might be a lower bound of how much persistence there has to be from parent to children. If we think genes are important for the outcome, and we know that genes are passed from parent to child, there might be some floor under which you can't go in how persistent outcomes are. That genes, in some sense, might put a floor in levels of persistence between children and between parents and children. So I think of that as a potential interpretation of this comment. And the second comment is something I disagree with, but you'll see why in a minute. To the extent, but it's common across many disciplines. To the extent that this genetic difference is the underlying cause of beta 1 or the intergenerational correlation, it may suggest a more limited role for policy. So that, I think you've heard that from across many disciplines, that if we think that something is quote unquote genetic, there's no policy response. So I think this is a particular place where gene-environment interactions come in and say that that's not true. So that's what I want to show evidence for. I'll skip this. I'm not in a room with economists. So I'll highlight just in some sense what to get ready for. There's a wave of this type of research coming, so I think it's at least worthy of some people having some knowledge of how these studies are conducted, what to believe and what not to believe in these studies, because it's coming. I'm going to show you some evidence of health and retirement study, but in many social science studies, so these first 4 were originally enrolled as social science studies, studying some things about lifetime income and health and retirement and so on, that have gone in some. You could think about these have all gone from relatively large social science studies with tens of thousands of people, tens of thousands of variables, to genetic studies that have a social science component, meaning that they still have 10,000 people, and they still have 10,000 social outcomes, but now they have millions of genetic variants in them. So just the size and the scope of these data sets, the first 4, are radically changing in some sense as we speak. So this is now available for anyone in the audience to use. This, I think, is going to be available either this week or next week, and these are coming, where there's genome-wide information in relatively large studies in the United States. This is an extreme version of this in the UK, where they're sending out 500,000 people with genetic information, large amounts of survey data. So just the scale and scope of this type of analysis is right in the middle of another sea change. We've already had several. Locally, the health insurance company has hundreds of thousands of people with all the health records and genotype information in it. 23andMe might then come on top of this and say, now, oh no, we have millions and millions of people with survey information, genotype information. So certainly understanding how the two exist and might say something, and how they might say something about social science research, I think is a fruitful area going forward. So I'll mostly skip this just to say that this isn't the first paper that did something that's going to do gene-environment interactions. Ben Dalton, who many of you know, and colleagues have a very similar, or have some similarities in this paper from Sociological Science a couple years ago, where one of the findings of interest was a gene-environment interaction, where the difference with what I'm going to show you is that their environment is the mother's schooling, and the G is basically the G I've already described, the education genetic score, and this is no evidence for gene-environment interaction in their particular case. Dan was here recently. and showed you things like this, which is a similar polygenic score of what I'm going to show you, a different survey. This is the Dunedin study. And this link between polygenic score and socioeconomic status as an adult, and this gradient as it moves across these figures is suggestive of gene-environment interaction, which is that the slope of this line seems to subtly change as you move from growing up in poorer families to middle-income families to high-income families. So that's some suggestive evidence of gene-environment interaction, that the effect of genes on outcomes seems to be somewhat shaped by, quote-unquote, the social environment. A little bit even more recent, a couple months ago in the economics realm, there's something similar. It's also health and retirement study. It's also, excuse me, examining years of schooling outcomes, and they also find evidence between the polygenic score I described, parents' background or family SES, and years of schooling. So there are papers that bring you up to what I'm going to describe now, which is that these three papers all do the same, something similar of what I'm going to show you here in the cartoon, which is we have measured child genotype, the polygenic score. We have something about the environment. And in these three cases, it was parents' education or SES. And we care about the main effects and their interaction. Okay. And these authors understand this issue that I'm going to highlight, which is that you have this lurking variable that makes what you care about hard to interpret, which is that what you care about is how is the interaction between parents' education, SES, or some other measure of environment and child's genotype, but you know there's this other thing that you don't measure, that's the thing, you don't measure this, that affects both the child's genotype through this transmission that you all know about, and affects the parent's outcome / the environment the child is raised in. So that's the problem. I'm going to address a possible answer to this, which is move this environment and measure it in a different way. So what I'm going to show you is a move to a more macro environment. So it's not about the family anymore, but it's about the state environment that children grow up in. So this will take just a second, because what I would like to have, well, I have a slide later to show this. What I'd like to have is something about the linkages between parents and children that are outside my family where I grow up. So basically, am I growing up in a state where there's really strong links between the outcomes of parents and child in the children, or am I growing up with states where that link is decoupled? So that's the measure of environment that I'm aiming for. So I'm going to be showing you evidence from the health and retirement study. The people in the study, I should say the children in the study, were born a long time ago. They were born from the 1920s to the 60s, and what I'm going to show you. So what I'd like to measure is, well, how easy was it for children to decouple their outcomes from their parents in the 1920s, 30s, 40s, 50s, 60s in the US across states? There's no data set that can tell you that. So what I'm going to do for now is to go within the HRS study itself, and so take me. So I was born in, take my dad. He was born in Florida in 1950. I'm going to take everyone in the HRS who was born in Florida in the decade of 1950s, but my dad. I'm going to run a regression that links the parents and children's education for everyone in Florida for that decade, and that's going to be my environmental measure. So I'll try to restate that. So see if that has sunk in the first time, and I'll restate that in just a moment. So the trick I'm trying to do is that here's the issue that I've described, and how I'm going to get around that issue is to do the following. Instead of this measure of environment, I'm going to create a different measure of environment, and now I'm going to ask the interactions between child's genotype, the polygenic score, and this other measure of environment. What has to be true for this to work is that the parent's genotype actually isn't moving kids into certain environments. Okay, so I'm going to show you some evidence for that that's consistent with that assumption. The data set I'm using is the health and retirement study. It's of this size. Many of you know it's been collected since 1992, and the genetic collection began in 2006. So if you're focusing on individuals who've been a genotype, you have about 12,000 observation. I'm going to use the polygenic score on only people of European ancestry. The The Manhattan plot, the tall buildings from earlier, is only from people with European ancestry. This, let's see, where is it? This. So everyone in this sample are people of European ancestry. So I'm going to take what you learn here and only apply it to the HRS sample who are also of European ancestry. So that gives me this sample from this. I also need for the people to tell me when they were born and what their parental education levels were. So that's going to move me from here to there. Questions about the sample selection? Yes?
It sounds like when you were earlier, smaller, samples, and you got false positives.
Well, the whole strategy in the false positive regime was different. So when the false positive, so the old paradigm where there was a candidate gene approach where you focused on a couple areas of the genome and you asked whether people differed based on these genetic measures, those were almost entirely done in sample sizes of hundreds or thousands, or small thousands. So the likelihood of false positives in samples of hundreds or thousands is really high. The only information I'm taking here on the genetic side is from the sample of 400,000, where that came from the Manhattan plot I just showed. So it's not to say that I can rule out false positives, but the information I'm using has first come from a sample of over 300,000 people and been replicated in the second sample, the holdout sample of 100,000. So it survived that, and then it also will predict in my sample. So, but I can't say letter to letter. I can't say that whether or not I'm using some false positives or not. It's still a concern. Yeah. Yep.
Is there a reason why the moderation effect is one way and not the other way? Right. So.
You want this one?
Yeah. Right. The environment be moderating the affected child genotype.
This is. I think your question is just the fault of my own cartoonish arrows here, which is what I want to say that I'm doing is interacting. So there's a main effect of the environment that you're going to see, a main effect, so that's this, a main effect of child genotype, and I'm also going to include an interaction. I think I'm interpreting what you're saying is that this red arrow could be pointing the other direction. So that's true. So the statistical analysis I'm doing is two main effects of an interaction. I think you can interpret it as whether the environment is being moderated by the genotype or the opposite. I can't tell you which way to interpret that. So keep that in mind before you see the results. Any other questions? So the reason I thought of this particular environment is what many of you have seen from Chetty, which is that in the United States, there's tremendous spatial and temporal differences in the environmental context. So mine is similar to this. I'm taking states and decades of birth cohorts and asking whether some places in the country have higher versus lower educational mobility, and that's my environmental exposure. This type of variation is my environmental exposure. Of course, what I would love to have happened is to have this map available for the United States going back to 1920. That's not currently available. So that's why I've had to go inside the HRS itself and recreate this type of map at the state by decade level only within sample. That's where I've run the 20,000 regressions where I take my dad and I take him out of the analysis. I take everyone who's from Florida, born between 1950 and 60, and run a regression between parents' education and child's education and get a coefficient. And that's how I populate these states. So I've said that twice now. Is there any questions remaining about how I'm actually calculating the environment? Like another try at that? Okay, so summing up what I'm about to do, so one, how are we measuring genetic inheritance? That's through the polygenic score that I've walked you through. Measuring environments that matter. The main movement I'm making is from other literature is the focus on families and parental investments to macro environments, so state levels of educational mobility. And then the research design considerations I'm proposing and promoting is that when you're examining gene-environment interaction, you're worried about what's termed gene-environment correlation, or you're worried about this. You're worried about parental genotype affecting both, which you know is true, the child genotype and the parental education, and therefore creating a correlation between genes and environment. So in the literature, that's called gene-environment correlation. And you want a clean separation between the environment that you're measuring and the genetics you're measuring. What's the HRS sample look like? So I'm going to be focusing on years of schooling. These cohorts here were born, like I was saying, between 1924 and 1963. Just a couple. So I'm only going to show you one of the analysis, but I've done the analyses based on different ways of measuring fathers and child's years of schooling. Is it a rank? Is it a standardized years of schooling? Is it just levels of years of schooling and so on? I'm going to show you a polygenic score. I'm going to show you a little bit of evidence across a region of the country where you were born. And I've also measured mobility a bunch of ways. And it's probably not important for this talk, but I measured it 4 ways. These are all highly correlated, so the results I'm going to show you aren't too sensitive to the different types of educational mobility you might measure so far from what I've. So let's interrogate this a little bit. What I need is that the parent's genotype is not sorting children into places that have high mobility versus low mobility. So what I'm going to first show you is that the child's educational score, the polygenic score, when I run a regression where the outcome is state-level educational mobility, there's no correlation between the child's educational score and the educational mobility of the state that he or she is born in. I found that somewhat surprising. It's certainly good for my research design, but I also found it surprising. One post hoc thought is that in the 1920s, 1930s, maybe we believe this to be more true than we would believe it today, that parents aren't sorting into states based on educational mobility in the 20s and 30s. That's post hoc. I think this is not crazy in that you see that people who are born in the South are born into states with much lower mobility than in the Northeast, so that seems like what you would expect. So that's true in the data, and the mobility rates are changing across birth cohorts. But the main point here is that I'm showing you some evidence consistent with this, that there doesn't seem to be child genotype correlations with environment. I can show you that across the whole distribution. So here I plotted the polygenic risk score, the polygenic score, based on whether you're in a high mobility state or a low mobility state. Your eyes might detect a little bit of difference. The stats don't in this particular case. I'll just frame that amount of difference. So I've taken this and pushed it up here to the top, so you can compare what I see for high mobility states versus low mobility states versus just men and women in the sample. This is also not statistically different from one another. And then something that is statistically different from one another, something that is statistically different from one another is whether your own father is high education or low education. So there you see something that maybe your eyes see has larger differences, and certainly there's statistical difference between people who have highly educated fathers and not highly educated fathers. So this is all evidence consistent with no gene-environment correlation or limited gene-environment correlation. So questions about that before I move on? So the main analysis then is to tie genes and environment to years of schooling. So here's the mobility measure that I was describing. Here's the polygenic score. I'm predicting years of schooling for the child. So to interpret this, a standard deviation increase in the polygenic score is associated with a 0.6 years of schooling increase. And I've already mentioned this, that the polygenic score itself has an R squared in the sample of about 7% of years of schooling, the whole sample. This is just descriptively just to compare that to years of schooling of dad and mom and together. This has all been done in the literature before. I think what's interesting now, just from an interpretation standpoint, is that this regression here, when you say that a child's educational polygenic score, when you increase it by a standard deviation, that child is predicted to have almost 2/3 of a year more of schooling. Part of what's happening here is that you're tagging that this child has parents who are highly educated, right? So you can see that a little bit from this column to this column. So this column is exactly this regression, but when I've added parents' years of schooling, and you see some attenuation in the regression because I interpret this as evidence that this is both child's genetic score being associated with years of schooling, but also that children with high genetic scores have parents who went to school longer. Okay, yeah?
Assuming these aren't standardized, like you can't compare coefficients because,
I think you can. Why?
well, I guess because I was looking at the R square, it gets much larger when you include father years of schooling, but then the coefficient on that is so much smaller. Because I was first looking at how the educational genetics score is almost double or triple the coefficient on father years of schooling in the final.
Oh, so yeah, so you're right. Let me go back to your main point. Standardized, I should tell you what units these are in. So you're right that they're not, I thought you meant just across columns, but this is years of schooling. So 1 additional years of schooling. It's not standardized. So that's just to clarify what you said. So that's right. So this is, when you're comparing it, you're comparing a standard deviation difference in the genetic score with a 1-year difference in father's schooling and a 1-year difference in mother's schooling. Just to, that's your first point. Does that change your second point, though?
No, I just clarified.
Okay. Any other questions? Yeah.
There are some families with single mother households, right?
Yeah. So,
Are your kids, I mean, what do you think about that?
so that's a possibility. So this is, I should make clear that this is all the child here, which
Question? I'm so sorry, I couldn't quite.
there's a question of missingness in parents' education. Is that how to interpret your question? Like, where's missingness coming from? What about children who have a single parent growing up? So this is, I should just say, I've taken all reports that the child makes on fathers and mothers schooling, regardless of residence. And that's what I have. And you have that over time. And I'll also just say that this is not a contemporary population. It's 1920, 1930. The levels of single parenthood are much different than, although there are some. So that's a partial. I should. So what I've just learned here is I should do a better job of making all the columns the same and thinking more about single parent families. Point taken. That's right. So let me go to the main set of results, which is I'm going to take a column like this and add a gene-environment interaction. So questions before I do that? No. Okay, so here's what you just saw, and here's the gene-environment interaction, which is to say that as you move around the state level education and mobility, you have a difference in how predictive the genetic score is. So in particular, going from no mobility to high mobility reduces the prediction of the genetic score by this amount. So there's probably many ways of saying this, but if you're in the least mobile place, a standard deviation increase in your genetic score has this size effect, years of schooling, so 1.1 years of schooling difference. As you move up to the most highly mobile, the places where child and parents' education is decoupled the most, that's the most highly mobile place, you knock that down substantially. So you can subtract that from that. Questions about that interpretation before I move on? So the main finding, I mean, there's several ways of saying this, the same thing, which is that the genetic score is less predictive in states with high educational mobility. So okay, so I'll stop there. Questions about that or other interpretations? So I'm not sure. So I think you know where I'm going, but now it's less clear though whether we should focus mostly on this column, which I just described, or if we want to then add parent schooling. Okay, because parent schooling, we know from before that part of this effect, before I do the interaction, is tagging that people with high genetic scores for education have parents who are highly educated, and that has lifelong effects of the child. So that would be one reason to cut that off and control for it. But on the other hand, this is the genetic score, and I'm examining gene-environment interactions. So this is what's given to you at conception. And the mobility measure is what's given to you at birth. This is a state of birth measure. So potentially we want to focus on this column. But I'm showing you both this column, and this is the column where I've added mom's schooling and dad's schooling. You see the same style of findings where in lowest mobility states you have a much tighter link between the genetic score and outcomes, and that's substantially reduced in the states that have the highest educational mobility. Yeah?
I'm trying to understand the fact that the R squared, so the first, there's a big difference in R squareds between the first two and the third, fourth columns, but between the columns 1 and 2 and 3 and 4, they're almost identical.
It's parent schooling. I mean, parent schooling is a substantial explainer of child schooling. So that's what's different about these two is you have the parent schooling in them.
Actually, that I got.
Oh.
I was wondering about the fact that they're so similar otherwise.
Oh, the additional explanation of the interaction, you mean, in terms of R squared? Yeah.
But it doesn't seem to make very much impact on the R squared.
That's right. So let me interpret this in two ways. One is the R square way, which is how much variance are you explaining in the population by adding that variable. Not so much, and that's what you're describing. An alternative view is if you think that I'm showing some causal effects, then it's saying something about environments that are not in this data. So for example, as we change mobility across time or across states, this is telling you something about predicting how genetic scores are going to be predictive or not predictive of the outcome. So I think they're both important, R squared and estimating causal effects, but they tell you different things, I guess. So definitely right that the explanatory power is not going up by a lot. But if you believe this to be a causal effect, then you might have learned something maybe about how mobility states with high versus low mobility operate. Meaning part of an explanation, part of why a state that has high mobility, part of why they have high mobility is because somehow they're able to decouple the genetic advantage that's passed from parent to child. How they can do that would be next steps. This is a question of, is it true? Does it seem to be true that places that have high educational mobility are successful or not successful in decoupling this genetic transmission? Because going back to some of the slides before, this now suggests some policy potential where that and it suggests what we're capturing by the intergenerational coefficient, and that it's not hardwired. It's not all genetics that's affecting the next generation. That it can be moderated by environment. Tom, did you have a question? Or
Yeah, I was wondering if you thought at all about issues related to measurement error and the mobility measure. Because it would seem to me that the state level measure might be plagued by considerable measurement error in terms of the environment people actually experience.
Yeah.
And so one, if you think that's true, then thinking about what implications that might have with this pattern of results. Because the own effects and interactive effects of the mobility measures are fairly large. And if we thought there were substantial measurement error, and that measurement error were classically additive, like these are attenuated estimates,
Yep.
perhaps?
Mm-hmm.
So just curious what your thoughts on that.
That's my first thought, which is that this is too small. Well, you're saying that they're attenuated. Whether it's more complicated than that, so I'll spin the story of where it gets more complicated, which is mostly reminding you of where the mobility measures come from, which is it's within sample. So I grab everyone in Florida born between 1950 and 60 who happen to be in the HRS, and I ask how tightly linked parents are to their children for that subsample. I mean, those are all. I've capped it so that all of those subsamples are number in the hundreds, but the question that you're asking is that a representative mobility measure for that state, for that decade? And that's where the precise measurement error becomes less clear, whether it's just classical and noise, and this might be attenuated, or might this be systematic, that I'm capturing people that are different than the general population in that state, in that decade. So my plan is, I won't be able to tell you the solution, but my plan is I can take the characteristics of these people and when they were born and where they were born and map them into the census and ask, like, how weird are these people who I'm finding in the HRS versus the people who were born in the same state and in the same year. And so that's what I've done with other samples. So in a different sample, it didn't matter there, but I don't know if it's going to matter here or not. But you're right that in order to mess things up in the other way, it would have to be systematic about some states, that have certain mobility patterns that I'm, always underestimating mobility in some states, or maybe always overestimating it in another state. So that's the concern, I think. Other questions? Yep. Yeah. Do you want to answer this precisely, or do you want me to vaguely answer it? No, not Yelena. Ben knows this. I'm putting Ben on the spot. So Ben has a paper with David Rakoff that using the HRS data that asks about genetic clustering in the states. It's unfair to him to say, like, what were your results from this one of your 100 papers, like right now? So that's something that is knowable, but I'm not going to be able to answer it for you now. A small answer is that what's hidden in these regressions are the polygenic, what's hidden in these or not shown in these regressions is that I have 10 principal components for all the people, so that their genotype outside of the polygenic score is controlled. But whether there's tremendous clustering at the polygenic score level across states and across years, Ben, are you going to come to my rescue?
So one weird thing about the HRS, they have
Can you talk a little louder?
yeah. So the HRS samples metropolitan. They sample basically I forget the exact unit, but some kind of census area, and they don't have a sample in every state. So to the extent that you have people born in every state, some states are only represented because people born in state X move to state Y, where there's an HRS sampling zone. So I'm not even so that to me would make this kind of question about that just really kind of complicates the whole thing in a way that maybe is worth.
Yeah. So it's certainly it's good for this isn't the answer to your question, but in the end, I'm going to go back to see how much I should and can rely on this, which is that the correlations in the state, whatever the sampling whatever's going on at the sampling level, in the end, if I ask what's the correlation between the state mobility that I was born into and my genotype, not zero. Like I can rule out almost everything here. So that's not obvious from the sampling design that Ben was describing, but it happens to be true, and it also happens to be true that other things we think should be true seem to be true in the data. Born in the South means that the mobility measure that I'm constructing in sample for those who are born in the South, you have lower mobility than if you're born in the New England and so on. So that's at least a plausibility point or a sanity check. So I've done a little bit more probing of the results. So I do have these birth regions. I thought I might get something more interesting in the South than some other places. It's possible I just don't have the power to detect differences in gene-environment interactions for people born in the South versus other regions. But so far, I haven't found anything measurable by South, Northeast, West, or Midwest. I don't call this suggestive, but if I just split the sample between men and women in the HRS, and this is a consolidated version of these last slides. Here's the gene environment and gene environment effect. You can, if you squint maybe. So this I'll first say these aren't statistically significant coefficients. I've split the sample not quite in half, so my power to detect effects is quite a bit smaller. But if you want to maybe overinterpret these, you see that in the states that have the lowest mobility, for the same increase in genotype, males get a bigger educational advantage than women. But also, as you move mobility, that advantage is more substantially reduced. I'd be interested in others' suggestions of other points in the data that you might be interested in seeing. Just as one other direction one could go is that the health and retirement study, so far I've almost used it as a study of young people because my outcome is years of schooling, but this is exactly not what health and retirement study is. Everyone in the study is 50 years or older, so I can see over the life course how these effects work out. So just as an example, I can when people in their 60s and 70s, I can ask about their accumulated wealth. and first show that the education genetic score is highly predictive, even accounting for education itself of wealth, and you can maybe benchmark that against a year of schooling of dad, a year of schooling of mom. These are log points, so this is a 1 standard deviation difference in educational genetic score is a 35% or so difference in accumulated wealth versus a 1 year dad schooling is about 7% and so on. The gene-environment interaction works in the same direction in this 5th column, where growing up or being born in this highly mobile state versus low mobility state has this longer reach into much later adulthood, where it depends both on your genotype in predicting your accumulated wealth, but also in the environment that you were born in a similar way. And I can show you a picture of that, which is you see this crossover. I've carved up mobility into 4 quartiles and run the regression. You can think of this as running 4 regressions, and you can see in the least mobile quartiles, the first and the second, you have this slope versus the third and the fourth, you have that crossover. So that's mimicking this negative coefficient. And this is partly I was analyzing some nonlinearities in mobility, asking as you move mobility up and down whether it seems to be really important to be in the bottom or the top mobility. This seems like there's just this carving around the midpoint that really matters of the mobility. So just to give you a scale, these are by the year 2008 or so in the sample, accumulated wealth, 500,000 million, as in a million and a half as you move the polygenic score around, and then as you move the mobility around. Questions about that? Yeah. Oh, yep.
So one thing I was thinking here is one way that genes can be related to both the environmental conditions and educational attainment is through the health outcomes.
Mm-hmm.
Presuming this kind of study you would have that. Does if you look at the insurance provisions that people have, or,
Mm-hmm.
or some sort of health mediators, or the provisions of healthcare in those states. I'm wondering if maybe what's going on is that the educational equity, the social mobility is picking up a whole lot of other aspects of equitable social provisions.
That's right.
Doing some sort of work here, right?
That's right. So I guess I think of this in a couple steps. One is how convinced people are on what I'm showing. I take your questions as, well, maybe I'm somewhat convinced about what you're showing. Let's think about the mechanisms of what it is different about states that have high measured educational mobility versus low, and what pathways might. So that, I think, is open to you and anyone else to say, if you believe this fact, then HRS is an incredibly rich data set to ask how these things are happening over a life course. I guess I would go the other direction also, in that before I got to wealth, the outcome of interest was years of schooling. So that's going back to age 20 to age 22. And that would be better, I think. So asking how states with high versus low education mobility produce these gene-environment interactions between the polygenic score and for years of schooling, how that affects young children in middle school and so on, that needs a different sample. The HRS won't be able to tell you anything about that because we pick all these people up when they're 50. So that's all to say. Another data set called the AdHealth that Ben works with, and that will be widely available soon, you might be able to start picking out where in these educational trajectories social mobility seems to matter a lot, or what are the mechanisms of these broader social mobility measures as we work through the educational system and so on. So I think those are 3 papers you could do, at least. Yeah, Yelena?
Quick little bit on this interaction. Should we assume the simple slopes are significant for all of those lines?
No. This is maybe a failed test of a lot of nonlinearity. I thought what was going to happen maybe was that across. So this is assuming linearity in the effects of mobility on the outcome. I thought, well, maybe there's some thresholds that really very high mobility places are different than moderately high mobility places. And this is more consistent with two types of places, low mobility and high mobility places, because the first and the second quartile hug each other, and those aren't statistically significant differences, and the third and fourth quartile hug each other a lot.
Okay, but let's collapse it.
Yep.
Are there still simple slopes significant for collapse versus second and third?
Yes.
This is not a flat line statistically.
You mean the difference isn't flat? Yeah.
Is the relationship between polygenic scores and wealth significant for the first and second? Is that slope significant?
I don't think I can tell. I think I'm almost positive it is. I don't think I have a table to back that up with me.
Okay, all right. So what's interesting is you're actually interpreting environment as a moderator.
Mm-hmm.
Even though your graph was showing GWAS polygenic score as a moderator.
Mm-hmm.
Do you know what this looks like when you flip it around? When the environment is at the bottom?
Right.
And then the last question I'm curious is, what's the distribution of your polygenic score? Does it really go to -4 and +4 standard deviation?
I'm sure you, so I don't think this is going to, so this is the score, but is this censored? I mean, partly you're asking how many people are here? Yeah.
I'm just trying to get a sense of, what this, because it's,
Yeah,
my graphing is so wide when most people fall within -2 and 2.
that's right. Let me think of if I know that answer off the top, or so I take part of your point as doing some sensitivity analysis of outliers, but whether I can rule that out here, I don't think I can. I mean, certainly the bulk of the people are between these two, and I've got to imagine that these results aren't so contaminated by outliers, because everyone's here, and I'm just testing for differences in those two slopes. But
I'm not worried about outliers. I'm trying to flip this around.
I know.
If polygenic score was a moderator, what that look like, and do we have decoupling of environment and outcome?
That's a great point. I don't have that with me. Yeah. Sorry, Ben. I'm just working through. Are any of my slides close to this? I think the answer is no. Sorry. Yeah. So the answer is yes, but this is a partial answer to your question. Versus 1920, the birth years 1925 to 1934 or so, this is the answer versus that. So mobility is increasing by those magnitudes over time.
So the reason I'm wondering is, I have a paper.
Yeah.
To be in this sample, you had to live until 2006, and
Yeah.
obviously you were born in 1920, lived in 2006, and then Carter passed in 1950.
Yep.
So I'm just wondering about the mobility coefficients you have in early birth reports, whether he's looking at 1935 to 45, might not produce some of that.
Perfectly fair point. I don't have that. You're partly, I mean, I'm trying to think of how to quickly address this, because I've done a bunch of cuts that you're not seeing. I don't think I've split it up into, say, birth decades and asking whether it's really different. That's a good comment. I mean, a lot of these I'd have to be pretty unlucky, but I don't know if I'm lucky or not on this. Oh, so one small thing, which is not going to solve all this. The mobility measures, you don't have to live to be genotyped to be in the mobility measurement, right? You just have to be in the HRS.
There is still, I mean, there's also the other paper about the HRS, about mortality selection just in general in the HRS.
Yeah.
Might still apply, but
That's exactly right. So I'm part of the way free of your comment, but not all, certainly not all the way. That's right. So yeah, question? Like the main one, yeah.
The coefficient on mobility is significant when you include parents' years of schooling.
Yeah.
And I was wondering if maybe it was just a measure of the general SES of
So I have, and so here what I need is an environmental hammer that will show up in a data set like this. And you're exactly right. So I've done in two directions. One, is it really mobility or is it something else at the state level? So I'm not going to show you this, but I have results somewhere where I include state fixed effects, right? So that's somewhat better about pinpointing if it's something about these states in general versus mobility in particular, but not perfect. I can certainly include something like average years of schooling or average expenditures at the state level. And the real issue is that you don't get this hammer at all. Like state spending in these birth cohorts, average years of schooling by the parents just don't affect children's outcomes as much in this data. So I can't rule out that they matter, but I can't detect that they matter either. Other questions? Yeah,
Sorry,
sure.
I don't really ask methodological questions so much. But something that I think is important with all of this is that when you set it up earlier, talking about, in some economics literature, people talk about a genetic floor, and then sort of the interaction was sometimes it was being interpreted as moderation of the environmental effect, of the environment moderating the genetic effect, which I thought would have made more sense given the setup. And I also thought that maybe more consulting of the literature about like the social construction of ability, might contribute to, I thought it was a priori reason to expect what you're finding.
The interaction?
So like that reasoning meaning that ability is something that we construct, and in places where there's more higher rates of educational mobility, we might expect those to be places where sort of the way that we brand kids are maybe less sticky in those places. Maybe that's why they're more educationally mobile. I guess.
So what I'm going to say is not against your story, but mobility here is the linking or delinking between parents and children, not necessarily how we construct. I mean, it's not way far from what you're saying, but it's a little bit more precise. It's the construction of ability, maybe not based on the child's performance or what we believe about the child, but it's whether or not it's linked to the parent themselves. So it's not against what you're saying. Like you could have an idea that we construct children's ability when we just look at the parents and say that's the child's ability. We look at the parents, if they're highly educated, then the child, we've constructed them to have high ability, right?
Well, I also mean like the way that we interpret, like what we mean by ability changes from place to place, right? Like how we think about, what signals ability changes from place to place, right? So we would expect the environment to moderate that relationship between, parents and children to some extent, right? Like, I guess I just thought that there's sort of another literature that talks about the reason why we might not expect much of a genetic floor, and that's, in response to that sort of economic literature you're talking about.
Mm-hmm.
And I just thought that might be useful in terms of like, talking about why we might expect this environmental moderation.
I interpret part of your question and comment as a different way of framing where the floor comes from, which is that the reason you would see a floor is because teachers and society looks at the parent and says, that's what I believe is going to happen to the child, and therefore you create persistence where otherwise there might not be some, right?
But which is something that has been documented in a lot of ways, right? Like there's a lot of people who documented that process.
Yeah.
No one's really done much to document the process by which genes cause, like, I don't know, somebody which people being more stupid in certain places. Like, so I'm wondering why that literature isn't maybe focused on more.
It's a good comment. That would strengthen the paper. Thank you for that comment. So let me wrap up because I have a few more, just a few more minutes left. So one, I just remind you in part because it was, I talked about it for a minute or two, but it deserves more than that, which is there's a major shift in understanding genetic influences on social outcomes. Partly this is data that 10 years ago there was no data to do this. Now there's way too much data and we don't know what to do with it. Just the task is quite different now. I think this presents a major challenge to incorporating genetics in social science. So the current approach, and I put a little asterisk because this is just months, a couple years old, and who knows what's going to happen later, is to take the new paradigm, which is many genes, all of tiny effect, and to summarize those into what I was showing as a polygenic score into a quote-unquote genetic predisposition or genetic propensity. I think so this is now editorializing. I think a key point of integration with social science is what I'm showing, a gene environment. It doesn't have to be exactly what I'm doing, but gene environment interaction is one way to bridge social science and genetics. You ask both the main effects of the two phenomena and how and why and in what circumstances they might moderate one another or interact with one another. My particular flavor and what I push in particular for how one does this is to try to couch it in a research design that might allow some causal statements to be made. So I've spent some time in the problem gene environment interactions versus the many circumstances where genes and environment are highly correlated, and you'd like to, if possible, separate those two phenomenon. I've shown you a example that might fit that, which is that the state that children were born in the 20s, 30s, and 40s in the United States seems to be uncorrelated with their genetic score for education. That's not going to always be true. It seems to be true in these data. So I've focused on this particular case because I think it meets this E environment being exogenous, and that's what I argue. So, and I think that if you start believing gene-environment interactions are really important for these type of processes, I think it points us back to enriching models of intergenerational transmission to focus on those phenomenon. I'll end by also just restating that we're kind of at an intermediate point about we have all this genetic data. Right now we're summing across the whole genome. What that lacks is that the advantage is that we're able to measure something that's previously unmeasured, genotype across the whole genome and create one score that seems to be pretty powerful out of sample. The disadvantage, which is cut here, is that we don't know what those polygenic scores mean. So that's a slight disadvantage of knowing of a really important variable in your data set that you don't know how to, you don't know the meaning of it. But I think in additional gene environment studies, we can start uncovering what the meaning of these polygenic scores are and understanding why some environments seem to matter and in what cases. So thanks very much for your attention and your comments.